Hello and welcome everyone. In this podcast, we're going to begin our discussion of proteins. We're going to spend the next several classes talking about proteins. In this class, we're going to break it up into two large sections, which will probably end up being in two different podcasts. But first, we want to talk about amino acids. We want to talk about their structure. We want to talk about how they help determine protein structure. We want to talk about the classes of amino acids and how that helps determine how this protein will fold. And then we want to talk about this next large section where we talk about more specifically the protein and how it folds. We'll talk about the 4 different levels of protein folding, the importance of how proteins fold, and what happens when they don't fold properly, and that when these proteins fold, they will help form specific domains that are very important in specific functions of the overall protein. And then we'll end with a brief discussion, nothing too in depth, but some talk about protein families. Okay, let's begin our discussion of amino acids by drawing one. We've drawn one of these before, but let's do it again. On the left, we always draw the amino end. It has a positive charge, and then we draw the next carbon, and then we draw the last carbon, which forms a carboxyl group. On this position here is what we refer to as an R group, or a side chain. And we'll come back to these side chains in a little bit. I'm going to draw an amino acid next to it here. And again, this one also has an R group associated with it. And we'll talk about these R groups in more detail later, but what you should know for now is that these R groups are what characterize one amino acid versus another. So what happens here when these combine? It's a condensation reaction. And if you remember, in condensation reactions, a water molecule is lost. This amino acid's carboxyl group will now form a peptide bond with this amino acid's amino group. Alright, so what we see here, and I'm going to circle it here in blue, is the peptide bond. In between the carboxyl group of one amino acid and the amino group of the next amino acid is the peptide bond. Regardless of how big the protein is, you can always determine how many peptide bonds there are by counting the number of amino acids and subtracting 1. So we have 2 amino acids here. 2 subtract 1 tells us we have 1 amino acid. If we had 100 amino acids, we would have 99 peptide bonds. As amino acids are added, they're always going to be added to this side here. So the next amino acid might come along here. Like so, with this being the peptide bond. Again, we have 3 amino acids, so we have 2 peptide bonds. This protein will continue to grow until it reaches its very end. And we always write the final protein with the N terminus here on the left and the C terminus here on the right. So if you're ever given on a quiz or an exam a large protein, you should be able to identify the N terminus, the C terminus, the peptide bonds. Should be able to tell me how many amino acids are present and how many peptide bonds are present. Now, I want to talk about the variability of these proteins that are being formed. And let's just make up a protein. Let's say this is the beginning part of the protein insulin. So I'm going to write insulin down here. How does the cell know to make this very specific sequence for insulin? Ultimately, it comes from the fact that the DNA codes for it. The DNA determines which proteins are going to be made. And so maybe here is the gene for insulin. It's the insulin gene. And this insulin gene is in every single cell of our body. So in cells in our body where insulin is produced, like in the pancreas, every one of those cells will produce this exact same protein. There is zero variability. Again, because the DNA here tells it to do it that way. So to remind you, the DNA here is transcribed, and it's transcribed into an RNA structure. This RNA structure has a very similar sequence to the DNA. There is some variability, and you'll talk in great detail about it when you get to genetics, or if you've already had genetics, you've already spent a lot of time talking about it. But this is the RNA. And the RNA now will directly be translated into the protein. Again, the main point I'm trying to make here is this insulin gene, in every tissue where it's being produced, it's going to take this gene here, it's going to make the exact same RNA, and this RNA in all these cells will make the exact same protein. There is essentially no variability. I say essentially because later on, if you take advanced courses, you'll learn that there are some exceptions to that rule. But for right now, we're going to learn this rule that one gene will make this one protein, and they will be the same regardless of which cell they're in. And in most cases, between species, the difference in this protein isn't that great. When you have insulin in humans and you have insulin in mice, that insulin gene and that insulin protein that's made from that gene is about the same. Okay, now I want to spend more time talking about amino acids. Okay, before we get started talking about amino acids, I want to remind you what the basic structure of the amino acid is. And what I want to focus in on are these R groups here, these side chains. This backbone here is the same for every single amino acid. This is the thing that changes. So let's talk about these different side chains. These side chains can exist in one of 20 different forms. So there are 20 amino acids. Or another way you could say it, there are 20 side chains. We can characterize these side chains, these amino acids, in one of 4 groups. We have positively charged amino acids, we have negatively charged amino acids, and we have uncharged polar amino acids. So they still have a polar characteristic to them, but they don't have a charge. And then finally, we have nonpolar amino acids. Sometimes these nonpolar amino acids I'll refer to as hydrophobic amino acids, but it is better to call them nonpolar. So these are the polar ones and then the nonpolar. We have positively charged amino acids. We have negatively charged 5 of the uncharged polar, and then 10 nonpolar. Okay, right now what I'm going to do is I'm going to draw these different side chains for you. These are groups. What I want you to know, I'll write them down here, is I want you to know which group each amino acid falls into. So which group each amino acid is in. Is it a charged, positively charged? Is it a negatively charged? Is it uncharged polar, or is it nonpolar? You need to know that. When you look at an amino acid, you should be able to look at that amino acid and say which of these 3 it is. You may not look at that, you may not be able to tell me that it's a leucine, or it's a valine, or it's a glutamic acid. At some point you will, but for this class you don't need to know that. But you should be able to look at that amino acid, and not knowing its name, but you should know where it fits here. You should also know a little bit about their characteristics. In this sense, and we'll talk about this later on, but I'm just going to throw it out here so we don't forget. You should know where at in a folded protein are each of these amino acids. In general, and we'll talk more about it, nonpolar amino acids are in the middle of the folded protein, and the polar amino acids are more likely to be on the outside of that fully folded protein. Okay, let's quickly go ahead and look at these amino acids. Okay, so let's begin with these 3 positively charged amino acids. They include lysine, arginine, one of my favorite amino acids, and histidine. Lysine will have 4 carbons as this R group. I'm only drawing the R group because the backbone's the same. It will have 4 carbons followed with an amino group. And these carbons will have hydrogen on them. And they will have that positive charge. Arginine has an interesting structure. It has 3 carbons a nitrogen with a hydrogen on it, another carbon with an NH2 on it, and an NH2 on the other side. This also carries a positive charge. How could it not be one of your favorites? Look at it. It's an awesome amino acid. Histidine, maybe my least favorite to draw. It forms a ring-like structure. And this is where the positive charge is on it. So those are the positively charged amino acids. Next, let's talk about the two negatively charged amino acids, glutamic acid and aspartic acid. Glutamic acid has, as the R group, it has 3 carbons. Off this last one is a carboxyl group, like so, so it's negatively charged. Aspartic acid is very similar, except there are only 2 carbon groups, like so. Okay, so those are our 2 negatively charged amino acids. You should be able to recognize these as negatively charged amino acids, and you should know that the 2 negatively charged amino acids are glutamic acid and aspartic acid. Okay, next let's talk about our 5 uncharged polar. We have asparagine and glutamine. They're similar to the structures of aspartic acid and glutamic acid, with a noticeable difference. Just like with their acid counterparts, glutamine has 3 carbons from this, forming this R group. But instead of having that O there, it has this amino group. Asparagine, like aspartic acid, has 2 carbons forming this R group. I should have put hydrogens around those up there to be accurate. So again, they're very similar to each other, except the asparagine has 1 fewer carbons than the glutamine. Okay, the next 3 in this uncharged polar amino acid group include serine, threonine, and tyrosine. I lumped these 3 together because they all contain a hydroxyl group on their side chain. Serine looks like this, with a carbon with the 2 hydrogens and then an hydroxyl group added to it. Threonine is similar. It has a carbon with a hydrogen and then a hydroxyl group off of it, and then a methyl group off of the other end. Tyrosine is our first ring structured amino acid. It has a CH2 followed by a ring and a hydroxyl group down here. These are the last of the uncharged polar amino acids. Let's now look at the 10 nonpolar amino acid residues. Since we just looked at the ring structure of tyrosine, I want to start by looking at phenylalanine and tryptophan, one of my other favorites. Phenylalanine, for its R group, has a CH2 followed by this ring. Tryptophan is a difficult one to draw. Well, there's just a lot to it. It's a double ringed one. It's the only one that has a double ring. As I said before, tryptophan is the only one of the 3 ringed amino acid that has 2 rings to it. And so that's a good way to remember that structure. Okay, now I want to carry on. We still have 8 more nonpolar amino acids. The first one I start off with here is glycine. Glycine is the simplest of them all. It just contains a hydrogen. The next one I want to show is alanine. Alanine just has a methyl group, so it's also a fairly simple structure. Now remember, these are all nonpolar, so they would be typically found in the middle of a protein. The next one I want to talk about is valine. Valine contains 3 carbons, but they split off like so, with 2 methyl groups on the ends. Leucine is the next one I want to show. It's like valine, except it has an extra carbon. The last one I'm going to show in this group here is isoleucine. Like leucine, it also has 4 carbons. They're just arranged a little different. Alright, now we still have 3 more to go. 2 of them are often paired together because they have sulfurs in them. They're the only 2 with sulfurs in them. And they include the cysteine and methionine. So these you should recognize because they have the sulfur in them. Cysteine is a little simpler. It just has the CH2 with the sulfur and hydrogen atom added to it. Methionine is a little bit more complex. It's a little longer. It has these 2 carbons with a sulfur group next, followed by a methyl group. And unless I miscounted, I believe this is our last one, proline. Definitely in my top 3. Proline is a fun amino acid. And for this one, I do want to draw the backbone of the amino acid. And the reason I want to draw it is because it forms a ring structure with the amino group here. Areas with a lot of prolines typically tend to be very rigid parts of the protein, or less flexible. Now again, you're not going to have to draw these, but you should be able to recognize them and know which groups they belong to. Okay, that ends this podcast on amino acids. In the next podcast, we're going to talk about proteins and how proteins fold into functional structures.